Okay. Well, again, thank you so much for joining us and making the time to be with us on our second webinar. It's really exciting. There was a lot of good feedback from our first webinar, and so we're excited to be able to provide this night, and hopefully you find it helpful. Hopefully you find it practical as well. I just want to start by a welcome to country. I want to acknowledge the traditional custodians of the various lands on which we work and meet on today, and the Aboriginal and Torres Strait Islander people participating in this webinar. I pay my respects to elders past, present and emerging, and recognise and celebrate the diversity of Aboriginal peoples and their ongoing cultures and connections to the lands and waters of New South Wales. So like I mentioned before, if you do have any questions that come up, please type them into the chat and we'll try to get to them. But we do have a lot of information planned for tonight. So with that, I will hand over to Professor Simon Hawke, who's going to start off with a bit of an update on COVID-19 and MS.
Good evening, everyone. Thanks for coming along. Can you hear me? Yeah. So this, obviously when COVID-19 came out, we had no treatment for it. We were extremely concerned, and it did make a big difference, I think, to what drugs we were starting people off if they hadn't been on medication for MS, perhaps using more interferons. But given the fact that so many more people in Europe had developed COVID-19 compared to us, data became available, which clearly showed that in the majority of patients, even with the Delta variant, COVID-19 was not dramatically worse if you were on treatment for MS. There were some conflicting results for those people who were taking very strong immunomodulatory drugs such as ocrelizumab. And with ocrelizumab, the issue is that it would be hard to make good antibody responses during the infection, but as vaccination became available to make good antibody responses to the vaccine, to protect you against the infection. Having said that, of course, antibodies are only part of the immune system, and T cells are very important. T lymphocytes are very important for controlling viruses, and they are not reduced in people who are treated with ocrelizumab. It targets specifically the B cells. They would be reduced in the early stages in people who had been treated with alemtuzumab, Lemtrada, because there you wipe out the T and the B cell populations, but only transiently. And similarly, those people who have been treated with oral cladribine, there's less general reduction in the lymphocyte populations, but both the T and the B cell numbers do fall down to some degree. Anyway, so we now, of course, have some great vaccines. You always hear the bad thing about vaccines because all interventions really, any drug, even aspirin, paracetamol would have, potential side effects. So it's not surprising that some side effects have emerged. But just the speed at which these vaccines were produced was really quite extraordinary, I think, and augurs well, I think, for our defense against the next viral infection. We know now about monkeypox, but undoubtedly there will be more and more viruses. So we're it's a good sort of training in a sense, although the figures obviously with COVID-19 are horrific. If you look at the numbers, the hundreds of millions of people infected and over 6 million deaths that we know about to date, if you look at worldometer.com or the Johns Hopkins site. Anyway, so the next advance, and this again has come at an incredibly rapid rate, are these drugs that are now available to treat people who actually have COVID. So we hope that vaccination is going to be protective, but we have now available, and these have received emergency assent to be rolled out in the general population from the FDA and also by our TGA. So we have these antiviral drugs, and then we have now a new drug, which is very similar to the third drug, which can prevent the development of COVID-19. So going to the various medications. So the COVID-19 virus is an RNA virus. So we're made up of DNA and RNA. DNA is our genetic code, and we make proteins from the DNA via RNA. That's just normal processing going on in the cell, in each of our cells. Different cells make different proteins. And that's because they have different RNAs. And some viruses are DNA viruses, and they are viruses like herpes viruses, herpes simplex, or the shingles virus. They're DNA viruses. They tend to be much more complex structures, very complex. Whereas the RNA viruses are rather simple, often one or two short strands of the RNA. And what the virus does, it gets into your own cell and hijacks your machinery for turning RNA into proteins. So that's what happens. So the RNA gets into the cell, and for the COVID-19, it's getting in via the spike protein. And that was determined incredibly quickly. It's obviously not the only protein in the virus. There are a number of other proteins. But this is one of the major proteins that's on the surface. So if you see the COVID-19, you'll see it looks very spiky. And the spike protein interacts with proteins on our cells, specifically the angiotensin II receptor. And that's how it gets into the cell. And once in the cell, it gets into the area of the cell where RNA is being turned into protein, uses your own cell to produce proteins, and then that reassembles the virus into new, what we call virions, which is a single viral particle. And then that replicates very rapidly in the cell, and then the cell may die and it gets released. So you get millions of these little tiny spheres, spiky spheres, and that's what a virus is. And the key parts of the virus are the RNA, but particularly which proteins are produced by the virus. So if we go to the first of those drugs, molnupiravir, this is a trusted and tried mechanism for affecting how RNA is produced. So basically you put a rogue RNA molecule in, so when it's put into the enlarging RNA, it stops it replicating. Then we have another drug, Paxlovid, and that's a mixture of two different protease inhibitors. So these were developed incredibly quickly. They worked out that these agents stop the protease, and that's the viral enzyme which packages the virus into a new viral particle. So they're very specific in a sense. And then we have one of the first recombinant monoclonal antibodies. So naturally to infection, we make heaps of different antibodies to the spike protein. This is a single monoclonal antibody in a large amount, and it can be given after exposure and infection. Now, all of these drugs are treatments for acute virus infection, COVID-19 specifically. They need to be given within 3 or 4 days of getting it. So if you're on medication that is potentially going to dampen your immune system down, and the medications of all of the medications we're using to treat MS, it's the most dampening down one would be the ocrelizumab, and then perhaps the fingolimod, Gilenya. And so I think if you're concerned, if you got COVID-19, then there is a hotline that you should ring, and then there's in each health area there's a designated infectious disease person. And that's how it worked initially, but I think your GP can now probably prescribe a lot of these. So that's circumvented that step. And it's not something you can just go to the chemist and get, because, for instance, with Paxlovid, which is very, very effective at turning off viral replication, certain drugs mean that basically mean that you can't take it. So if you're on Tegretol, carbamazepine, even drugs like statins to lower cholesterol, you might have to stop them during the 5 days of treatment. Probably won't matter for some drugs, but say you had epilepsy and you're on Tegretol, you wouldn't be able to stop it. So you might have to use something else. No particular concerns with interactions with the sotrovimab. So that's also very effective. So that would potentially be an alternative. So I think my recommendation would be to go for Paxlovid or sotrovimab if you're concerned. Now, having said that, and I'm sure many of you have had COVID-19. I always ask people, have you had COVID-19? And it's, it's been so prevalent now. And in fact, there was a study done with the blood, just an unselected group of blood donors with the blood transfusion service. And over a 3-month period, I think there were 5,800 donors. And they could test against another protein, antibodies to another protein that can only be detectable in natural infection with the virus, against the nucleocapsid protein. So we've talked about the spike protein. This is another protein on the surface of the virus. And when they tested unselected blood donors using an antibody, or having an assay looking for antibodies against the nucleocapsid protein, 50% of donors were positive. So that means that of that group, half of blood donors had probably been infected. Many people had no idea that they'd been infected. So it is very, very prevalent. And I think it underlies the score that for most people, it's not that serious, the virus infection. But nevertheless, if you are significantly immunosuppressed, we do advise that you to get these other drugs. And then finally, we now have available, and this is really only for people who are extremely immunosuppressed, what we call pre-exposure prophylaxis. And these are 2 different monoclonal antibodies. So they have the same target, the spike protein, as sotrovimab. But this is not a cocktail, but they're 2 separate monoclonal antibodies. And you actually give it to people before they're infected. And it's given via intramuscular injection. And it's now available. And again, thinking that, well, most people don't have very severe infection. Is it necessary? Well, I would have to say that my MS expert colleagues in Australia are now giving this or offering it to people routinely if they're on ocrelizumab. We don't yet know how often you should give it. It lasts 6 months. You might have to have it every 6 months, because when you're on ocrelizumab, you don't produce B cells as soon as you start it, and they will only come back when you stop it. So that's a new type of preventative drug that seems relatively effective, although the data on how effective is a bit uncertain, but nevertheless, these antibodies are pretty safe. So I think that's the current situation with COVID-19 and MS, and certainly we can take some questions. Sorry, I've gone on way too long.
No, perfect timing.
Oh, good. Pure luck.
Yeah, if you did have any questions about that, do pop them up in the chat. Just oh, I'm still sharing my screen. I'll just stop sharing for a minute.
When you think how long it took to develop drugs to inhibit protease in HIV, it was like, 10 years. It's just incredible how quickly science is moving these days. Really, it's very encouraging, remarkable.
Yeah, absolutely. We don't have any questions about that just yet, but just to reiterate that if you haven't had COVID and you do test positive for COVID, a great step is to call your GP so that they can prescribe one of those antiviral therapies for you. And I think a few people have had that. So we'll move on to our next section, which is an overview of diets. So I will share my screen. Again, cute bacteria. So diets and MS. So before I give an overview on diets, a lot of people have asked the question, is there the best diet? What's the best diet for MS? What effect can it have on my MS disease activity? Does it help with any symptoms? So I want to give a little bit of an introduction to the gut microbiome, a very simple one, and also an overview of the main diets that you might be looking at. So I wonder, does anyone remember the Inner Health Plus ad with the, the high-pitched bacteria? So that's a good picture, basically. That is a good introduction to the fact that in our gut, we have millions of bacteria. So our gut microbiome. So I included these cute looking bacteria just so that you can have a visual of all of these. There's bacteria, there's fungi, there's viruses that are living in your gut. So when you think of MS, it is an autoimmune condition where the immune system attacks the brain and the spinal cord. Now, the gut, which includes the large intestine and the small intestine, is the largest immune organ in us. So each of us have millions of these microbes, bacteria, viruses, and fungi that live in our gut. And they have a relationship with our body, and they actually produce molecules that are beneficial for us. So some bacteria help us digest foods. You might hear that you have a lot of good bacteria. And so this is what we're talking about when we say good bacteria, that you have all of this living in what's called your gut microbiome. And this is what most of the bacteria are harmless as long as they remain on the inner wall of the intestine. And so more and more research is showing us that these bacteria help maintain our immune balance. And if you've heard of what's called the gut-brain axis, it's basically this connection between your external environment and the central nervous system. So it's the communication that happens in your body between your brain and your gut. So what does this mean for people with MS? Well, there's a growing body of research, and a lot more research is continuing to go into understanding the gut microbiome. But studies have shown that the gut microbiome of people with MS is different to people that don't have MS. So that can mean the diversity of bacteria is different. And I think I've also read the mass of bacteria is different. So that's really interesting because it makes us wonder, how can we change that and what can we do about that? So your gut microbiome is influenced not only by genetics, but largely by what we eat. And so, as I said earlier, that continued research means that we need to focus on, are there other ways that we can treat MS? And what is the relationship between diet, your gut microbiome, and MS disease activity and symptom management? So I'll give a little overview of a few of the common diets, and these are very simple. You'd have to look deeper into them, but there's the keto diet or ketogenic diet. Now, the main elements of this diet is that you avoid carbs and you consume more fats. And the idea of this is that your body will burn fat for energy, and your body goes into a state of ketosis by turning your fat into ketones. So there are a lot of low carb foods that you can eat, such as eggs, fish, beef, chicken, vegetables, fruits, nuts and seeds, dairy, and fats such as extra virgin olive oil and avocado oil. And some studies have shown that following a keto diet for people with MS has shown a reduction in neurological disability, fatigue and depression, and improved quality of life. So I will fly over these, but you can get, I'll send everything through later. So the WALS protocol. Now you might have heard of this one. This diet is a modified paleo diet, and it was developed by Dr. Terry Walls, who was diagnosed with relapsing remitting MS, and then it became secondary progressive MS. So she had good results on this diet and claims that it improved her mobility. So what is it? Basically, you're avoiding wheat, grains, legumes, dairy, and sugar, and you're consuming a lot more leafy greens, lots of fruits and vegetables, meats and fish, and foods that are high in omega-3 fatty acids. So again, the evidence with this is there's been a few small studies which have shown for people with MS that it has, following this diet, has reduced their fatigue. They have experienced an improvement in their quality of life. However, there have been studies that have also shown that following this diet might lead to you being deficient in a few different nutrients, such as vitamin B. Now, the next diet is the SWANK diet, which is also another, it's another common one that is often recommended for people with MS. So this diet was developed years and years ago. Basically, Dr. Swank, he recommended that his patients with MS consume low saturated fat diets, and he followed them up for 50 years. So it's not a very high quality study because he didn't have a control group, but he found that in the patients that consumed the least amount of saturated fat, that they were less likely to have exacerbations of MS, and they were more likely to ambulate. So on this diet, you're avoiding processed foods, saturated fats, and red meat. So no red meat for the first year. And you can Google how to follow these diets. And with this diet, lots of fruits and vegetables, nuts and seeds, whole grain breads, and rice and pasta. So the initial study was pretty subjective, but a randomized trial did see that following this diet also resulted in reductions in fatigue and improvements in quality of life for about half to 3/4 of the participants. Again, this is another diet that may result in some nutrient deficiencies. So there's another diet called the Overcoming MS diet. Now, this one was developed by Professor George Jelinek, and he was diagnosed with MS in 1999. And he currently also researches modifiable risk factors of MS. So there's no randomized controlled trial that validates this diet, but he does say that it is based on other scientific studies. Now, the main components of this diet is avoiding dairy and saturated fats, and focusing most of your diet on plant-based whole foods. So whole foods are just, one ingredient foods. So foods that are also rich in omega-3, grains, fish and seafood, and using alternatives instead of dairy. And the final diet that I want to give you an overview of is intermittent fasting. So this one's a little bit different. Basically, you're not necessarily changing what you eat, but you change when you eat. So you might eat for just a certain period of time from, say, 12 o'clock in the middle of the day until 8 o'clock at night, and then you don't have anything after 8 p.m. until lunchtime the next day. The main thing with intermittent fasting is that you want to maintain your proper nutrition, so you're still maintaining a balanced diet, but overall you're reducing the total food intake. And so the idea behind this diet is that the evidence that has come out from this diet is that it can induce healthy weight loss and also positively change your gut microbiome composition and have anti-inflammatory effects, as well as neuroprotective effects. So a lot more study is going into different diets, but I suppose the key takeaway is because we don't have a huge randomized control study, we can't say that one diet is the best diet for everyone with MS, but there's a few key elements that you can see from all of those diets. While there's been a lot of people have reported a reduction in fatigue, a reduction in depression, improved mobility. So these are some key takeaways, which I think are definitely practical things that that hopefully you can make doable in your own life. So reducing processed foods and eating whole foods. So what are processed foods? It's easy to say it, but just to break it down for you, this includes lots of cereals, bread, snacks, chips, sausage rolls, pies, pastries, sausages, ham, cakes and biscuits, soft drinks, microwave meals, or ready meals. So these sorts of things which are really easy for us to eat. And of course, when you're fatigued, it can be the easiest option, but these are highly processed foods. So also when you think about what to prepare for your meals, if you can minimize saturated fats, these are often in processed foods, and also increasing your fruit and vegetables. So I think the key encouragement is to try and make changes that you can be consistent with. Diet definitely has the potential to change your mood, your fatigue levels, and your immune system. And reducing your weight does also increase those benefits. But of course, try and make it a sustainable thing that you think that you can definitely change as a whole lifestyle change. So I hope that was practical. Of course, if you have any questions, please let me know. So I'll hand back over to Professor Hawke.
We do have a question, I think, from Prue Wilson.
I'll read the question out. So I contacted my GP clinic, reported I had tested positive to a rat and felt extremely unwell. The GP on duty did a short telephone consult and prescribed antiviral capsules. He sent the script to my chosen pharmacy, who delivered the medication within 2 hours. I feel very well served. I hope everyone has a similar or even better experience. The symptoms lasted nearly 2 weeks. Thanks for sharing that, Prue. That's really great to hear that they had a positive result for you.
That's really, really good. Yeah, look, I think I'd like to emphasize the importance of these diets, but I'd also like to say that a lot of research has got to be done to try and understand what's going on, I think. One of my scientific colleagues at the University of Sydney rang me up a month or two ago and said, hey, I've got these mice on a ketogenic diet and I can't induce the animal model of MS, EAE, which I thought was very interesting. It is pretty tough, the ketogenic diet, though, if you're going to be really rigorous about it. And but anyway, there's a lot more that we don't understand. Fascinating area. Okay, so now I think last time people wanted a little bit about progression in MS, and it's interesting how the concept of progression in MS is evolved. Now, when in the old days, when we didn't have MRI, we were pretty much based our diagnosis of MS and following people with MS based on relapses, we knew that people usually would start off with relapsing, remitting MS. And that at about 10 years from the diagnosis, about 50% of people would slowly get worse. So this is a concept of that sort of evolved from the days when we didn't really have very effective treatments. So what you'd have to say is that, well, a large amount of philosophical thought has gone in the scientific community to thinking perhaps progressive MS is actually a different disease. So somehow we have the inflammatory side of MS, and that's where the lymphocytes cause the lesions of MS. But then if people are slowly getting worse, maybe there's like a neurodegenerative phase of MS, which is different. Now, my view is that a lot of these separations of the different stages of MS is a figment, in a sense, of harping back to the era when we didn't really have a good idea of what was going on in the brain because MRI wasn't available. And what we know with MRI, and you all know that we do frequent MRIs, that it's quite the usual, not the exception, to see that the disease is active just on MRI and you have no idea that you've developed new lesions. So what that tells us is that irrespective of if you had an attack yourself, such as you went weak down one side or got some new sensory symptoms, had double vision or whatever, irrespective of those symptoms, the vast majority of people now, we assess the activity of their MS based on MRI. And excuse me, what you can tell with MRI, if you give contrast, and I don't usually ask for contrast, but I might in the first few MRIs, what you can see is that the recent lesions light up with the contrast because the blood-brain barrier is broken down where the immune system has caused an acute inflammatory area in the brain, and the old lesions don't. So you can use that to help make a diagnosis. Very useful because you can say that some lesions are old, some lesions are new. That fulfills the old criteria that you have to have an ongoing attack on the central nervous system by the immune system. But in terms of with our new treatments, if people are on treatment and we monitor them, we usually find that we don't see new lesions. Now, it has to be said that sometimes it's harder to see new lesions in the spinal cord because the spinal cord is really quite small. It's much easier in the brain. But on the other hand, if the immune system is still active, there's no reason why it would just cause a lesion in the spinal cord and not in the brain. So we use the MRI to help gauge if the treatment's working. So does progressive MS, a different disease, actually exist? And I think this is a difficult one because people who tend to get worse without having attacks of MS, you have to say, well, firstly, are they still having attacks of MS on MRI? And that's why they're slowly getting worse. Or if they don't have new lesions, is it possible to get worse irrespective of having new lesions? And I guess thinking about that from a philosophical point of view, you might think of somebody on a very powerful drug like Tysabri, natalizumab. And that blocks the entry of new lymphocytes into the central nervous system and completely stops new lesions developing. It's incredibly powerful. As we know, the problem is the JC virus, but it is very effective. And is it possible for somebody to get worse who's on Tysabri? And then I can turn around and say, well, actually, when they did clinical trials of Tysabri in people with more advanced MS, that didn't work. So I think the answer is yes, it is possible for people to get worse, even if they don't have new lesions. So the next question is why? And I think this is where we don't really know the answer. But it does tend to be people who have had quite a lot of damage to the motor pathways who will get worse. And my own view is that age and just getting older is probably a major factor in that. If you think about the motor pathway, so when you think of moving your right hand, you set up some electrical impulses in the motor cortex in the left side. And so that thought, if you like, is translated into electrical conduction down this tract. It's a bit like a cable going from the motor cortex, going down into the spinal cord, into the cervical spinal cord in your neck, and then going out of the spinal cord into the different nerves and then moving your hand. So if you've had a number of lesions that have been affected different parts of that same cable over the years, what that means is you'll have less, if you like, wires connecting you thinking of moving your arm and where it leaves and goes into the nerves, that the cable, overall, the motor cable will have less. And the same will go for the sensory cable, because that's how the nervous system is made up. We have different cables doing different things. A visual cable, of course, that's very simplistic, but in essence, I think what that means is normally, and I think we're all losing brain cells after the age of 25. I keep saying that, I'm sure I've said that to many people. Usually it's a very slow process. It doesn't actually matter if you lose 20% of your wires in the particular cable as you get older. But then if you've damaged the cable, or the cable has been damaged, I should say, by the MS, if you lose 20% of wires, that can make a big difference. So I think that is certainly one explanation why people who had quite a lot of damage can get worse. So that's on the one hand. On the other hand, there are some very rare people who have primary progressive MS. And I think we now understand that that's a very rare condition. And there are various types of primary progressive MS. And in those individuals, there doesn't seem to be a lot of inflammation going on. And there's even a type of MS where you can just have one lesion and people slowly get worse. So that's a condition called solitary sclerosis. And it's very rare, but it can affect younger people. So I think it is possible to have a single lesion and for that lesion to continue to be active and for people to slowly get worse. So, but that's really very rare. I think for 98% of people with MS, if you're turning off the inflammation effectively, and here we're so lucky in Australia that we can treat people aggressively if they seem to have very auto-aggressive immune systems, we can give the appropriate treatment, we can make sure it's working. So I think that I would be not so fearful, if I was starting out with MS in my early 20s now, as long as people are on very, treatment that's effective as measured by MRI, I think it's extremely unlikely that they are going to slowly progress like was quite common in the days before treatment. So I think it's a bit artificial to say, I've got relapsing remitting don't worry if, am I progressive? Have I got into that stage? Because I think if your immune system has been turned off from attacking your brain, you'll, if you do get worse, it's not going to be that quickly. So that's my view of progressive MS. Take a much more optimistic view. If you have any questions, please ask. So what's next, Karina?
Well, we do have one question
Yep.
that just asked, what's the latest research telling us about vitamin D deficiency in people with MS?
That's a very interesting question. Some of my colleagues at University of Sydney, they're photoimmunologists, and they're experts in how sunlight interplays with the immune system. So, of course, we know with MS that we've known this for many years, that there's this latitude gradient. So that means that MS is much more common the further away you go from the equator. That in itself is a very complex thing to try and understand fully, but it does seem that whether you're in the northern regions of Scotland, the Orkney Islands or the Faroe Islands, or in Tasmania, compared to other parts. So Dr. Hammond, my previous colleague who's now retired, he did the seminal experiments or work, epidemiological work during his PhD, showing that MS is much more common in Tasmania than in northern Queensland. So we have this, what we call latitude gradient in Australia. And the same goes in Europe and in the UK. It also occurs in North America, but there are some exceptions. So the obvious thing to explain that is sunlight exposure. Less sunlight exposure, the further away you go from the equator. So the next obvious thing to explain sunlight is vitamin D. However, it is, I think, because it's such a complex effect of sunlight on the immune system in the skin, vitamin D is just one aspect of it. And clinical trials of using vitamin D and showing that it has a very significant effect on slowing down disease progression or even relapses has been rather hard to show. Now we have a study in progress, the Prevent study. I think they may have stopped recruiting now. That's run out of Professor Taylor's group in the Menzies Institute in Hobart. And what that study did was treat people very early on. So they just had the first manifestation of MS, randomizing people to vitamin D or not. And we haven't got the results yet, but other studies have been done. And I think at best, vitamin D has a modest effect. So I think the bottom line about sunlight is that sunlight's important, but vitamin D is only one effect of sunlight because it's pretty clear that sunlight exposure, ultraviolet radiation, has profound effects on the immune system, irrespective of vitamin D. It has effects on antigen-presenting cells, on B lymphocytes, and they actually did a study in Perth using phototherapy in patients with early MS. And we contributed some scientific data to that. But yeah, so vitamin D is just one aspect. So I think it makes sense because it's safe and it's good for your bones to take vitamin D. But I think it's compared to disease modifying treatment, it's probably extremely weak. in terms of its effectiveness at stopping new lesions form. So that's my overview of vitamin D.
That's really interesting. We just have another question. Would it be beneficial for Linda to have Evishield because she hasn't had COVID and she's now on Kesimpta? So I think, I suppose, yes, the answer is yes. I suppose it's how should patients access it?
I think, I'm not sure, because well, yes, Kesimpta would be the same as ocrelizumab, of course. So I guess the criteria for it will be things like how immunosuppressed you are. So I guess, yes, that would be, certainly you could think of having it. Yeah, I think it's certainly something to consider, because Kesimpta, ofatumumab, is basically going to have the same effect on your immune system as ocrelizumab. So the answer is yes, and you probably would be able to get it. What we don't know is how effective it is. I think that's the problem, and you're probably going to have it every 6 months via intramuscular injection. But I think if we're being consistent, if you're on Kesimpta, yes, you should probably go onto it.
Yeah, and so should she see her GP to arrange that?
That's an interesting one. I think I've just started a prescription for the first person today, and I did notice he was here earlier, but I don't know whether he would like to comment. The forms that they've sent us out are fairly complex and a little bit confusing, I find, or I found when I was filling them out today. But it seems that you have to, your GP or I would have to fill these forms out, and then they get sent directly to the hospital pharmacy. Then the hospital pharmacy will dispense the vials of the Evishield, and then your GP gives it, because I wouldn't be giving an intramuscular injection into your bottom, I'm afraid. I might hit the sciatic nerve or do something stupid. So I think it's, yeah, look, your GP or your practice nurse would probably give it. So I think it's the actual mechanism of getting it is, because New South Wales is, that my colleagues in Queensland have been using it for much longer than us here, but that's how it has to be done. So we need to, if your GP can't do it, contact us and I'll get the forms, and we then have to get them. We can't give you the forms. It's very strange. I can't give you the forms. I can only give you a photocopy of the forms. The originals have to stay in your notes. And you then go to the pharmacy, take the vials, but I'm not sure what GPs are going to think about that. If you turn up and say, hey, I've got these vials of Evishield, do you mind giving it to me? So I'm not sure, but if you do, if you're having, yeah, it'd be interesting to hear your stories about what happens if you go to your GP and ask for it.
Yeah. We have another question from Nicole. I was diagnosed in 2009 with no new or active lesions since then. I'm now completely wheelchair bound. I'm on Aubagio. Do you think it's a waste of time taking it, as I'm thinking I've progressed to the next stage?
My view is that even if there's been a lot of damage to your nervous system and you're wheelchair bound, the question is, do we want any more lesions? So I think that, I mean, there have been 2 clinical trials which have shown some effectiveness in progressive MS, one with ocrelizumab and one with siponimod, which is very similar to Gilenya. Now, when you looked at those studies in detail, they were working in people who had new active inflammatory lesions. So they were progressing, but they had new lesions. So it was stopping the new lesions. So I think that even though you've had the disease for a long time and you're in a wheelchair, the next question is, should people stay on treatment for the rest of their lives of any type of MS? Not so much that I would say that it's not working, the Aubagio, but we would need to answer that by looking at MRI, because if you think you're getting worse, then are you getting new lesions? And my experience would be that most people who think they're getting worse are not getting new lesions. But I wouldn't take people off treatment because I wouldn't want new lesions to cause further damage, if you see what I mean. So this is totally against what I'm allowed to say. So, I have to say these disclaimers. This is not legal, okay? So sometimes I say, come and visit me in prison. But most of my colleagues would be prescribing disease-modifying therapy in people with fairly disabling MS because we don't want new lesions, but we're not allowed to prescribe it, but we do. So I think if it's been prescribed and you're tolerating it well, and there's you are getting worse, the first question is the drug working? Because Aubagio is a relatively weak agent. So that can probably be assessed by MRI, and you might do an MRI with contrast there, because you might want to know if there's active lesions. And if there were, I would probably switch it to something else or suggest that we switch to a stronger drug. If on the other hand, there were no new lesions, then the next question is age, because I think my own view is that we don't need to think of treatment lifelong, because once you get into your 70s, then your immune system isn't as active as it was. And that's why things like shingles is much more common over the age of 60, because your immune system, its effectiveness declines. This is irrespective of what medication you're on. And the same sort of thing goes with MS. So I think, we know from the natural history observation studies that were done, 30, 40 years ago, that usually the attacks of MS became less frequent as you got older. So I think when you get into your 70s, maybe there's a sort of a risk-benefit ratio. Maybe it's not a good idea to be on treatment. But certainly, I think if you came off treatment, you would want to monitor the disease with MRI to make certain that you weren't getting new lesions. So I think this is a concept now that many of my MS expert colleagues are saying, we should be thinking of taking people off medication once you've reached, and it's hard to know exactly how old your immune system is. So I think that, but I think certainly mid-70s and your 80s, you'd have to have a good reason for thinking that it's safe to keep people on medication. So that's the way I look at that.
Okay.
Okay, so I think we've pretty much covered everything now, haven't we?
We have. Thank you for sending in those questions. Yeah, that is everything. Is there anything else that you wanted to add, Professor?
Well, no, I did have a section on immunology, but that could take another hour. So I think maybe we should hold that one over for the next one.
Sounds good. Sounds like a plan.
And
so we will have another webinar. Sorry.
So I was going to, because I know many people have donated blood samples to our research. So I was just going to give sort of an overview there, but I think we don't have time now. So I think I've rabbited on too long. I'm sorry about that. But so
No,
I think
it's been really interesting.
I think what we've been talking about has been important.
Definitely.
Yeah.
Okay then. Well, thank you so much, everyone, for joining in. Thank you so much, Professor Simon, for that all of that interesting insight. It was really good. So we will have another webinar coming up in October, and we'll also be sending out an evaluation survey in November to ask what you think of these webinars and newsletters. So please keep an eye out for that. We would love to hear your feedback. Until next time, thank you, everyone. Bye.